Hello, everyone. So I'm very well aware that a lot of you have a lot of experience doing this. But the purpose of our work, when we published this paper, we started working with this with some of the people in this room a long time ago, a couple of years ago, was to provide a beginner's guide, something that if you don't know any Unix, if putting a password into your terminal and expecting something to show up was a bit clueless, you end up getting like, why can't I see my password there? This is the place where you need to start, okay? So why do we want to manually curate libraries? So there's fantastic work being done by all the automatic tools that we know, some of them that we've shown today. But there's still a lot of interest in perhaps understanding more about the structures of these individual TEs. And sometimes we see that these automatic methods do not achieve the full length of the TEs. We might want to really get into the nitty-gritty of the TE diversity within a given organism. And also we might want to having full length, pre-decorated annotated libraries, we might be able to be a little bit more confident about the TE classification. So these are the reasons why we want a manually curated library. In my case, I work with a very obscure parasite, and there's really not a lot of data out there, so I had to start from scratch. And I thought that spending time curating these things was really, really cool. And I now have a very a shamefully very close relationship with the TEs and these parasites, and I know them really very well. But then you can be smug about it in this audience. So this is what we came up with, and I have to highlight that none of these people knew each other before we got into writing this paper. It was all an effort of getting people together through Slack, talking freely about what we wanted to do on Slack, having video calls, and that's how Clement, Rory, Agustin, who's a student of mine in Uruguay, Valentina, Aaron, and myself got together to write this guide. No, I have to say this was the easiest paper I had to handle in my life. It was extremely agreeable to work with all these people. Everybody was willing to help, and there was no rivalries when the order of authors came out. In fact, everybody was surprised, was even given a spot. So it was a fantastic thing to deal with. I would recommend any of these people to you as a co-authors anytime. Right, so the next few slides, I'm going to summarize the process of what our proposed pipeline does. And then I'm going to do some worked examples, including some very basic Unix commands. So if you were puzzled before, afraid not, you'll be taking one step at a time. Okay. So the first thing we need to do a manual curation is to find query sequences. As you saw earlier, Repeat Masker uses a library to start masking the genome. So we're going to do the same. So in our experience, we have used Repeat Modeler 2 and EDTA, but there's a bunch of other tools there. And they will, the result will come up in the format of a multi-FASTA file where you have the sequences that represent prospective feeds. Right. And then hopefully you'll have the genome in which these TE libraries were formed from. Sometimes you may not have a library, in which case you can have round pathways in which you can get there. You can look for example, domains that you know are in transposable elements, like reverse transcriptases in the genome, and there are ways in which you can do that, although that's probably a bit more complicated. Okay, yeah. Oh, and the rest of the tools you can find in TEhub. And they're tools. Okay, so our little the first thing we do in our pipeline is just a bash script that I wrote myself, and it just takes a query sequence that is just a FASTA file. It could be one of these proposed families that you got from your repeat modeler run, for example. It will get this sequence, it will blast it against the genome, it will find its locations, and it has the option of extend to the side. So my experience, and it was shared with a lot of the co-authors, was that most of the time this potential putative families are quite short, and there's a little bit more to extend there to find the actual boundaries of that TE insertion. So there's a variable extension flag in the script. Then we lift those sequences from the genome, and there is an option to align them. You can align them manually, or you can run another version of this script, which is called make align from BLAST, and it will run a mapped alignment. And after that, you need to do a manual curation, which is, of course, a little bit of a death trap. And this is the main thing that I'm going to explain to you today, because at the same time that we were developing this, writing this manuscript, a colleague in my department wrote CI Align, which some of you have heard of. So I'm going to show you the latest developments on that and some parameters that have been optimized for TEs. So now I'm going to just run 2 of the scripts for you, and you can follow this in your tutorial. First of all, this is, for those who first start in Unix, the folder navigation is not perhaps intuitive, but you can think of it, this is just my folder in my computer, and this is the same structure that you have in your folders in the VMs. So you have one folder that is called TE man and not, and within that folder you have a bin where all the scripts are, and then there's other bunches. So this is just a clone from the GitHub page. Includes, it has the manuscript in it in case you want to cite it when you do your work. In another folder, in the tutorial data folder, that's where you have the chromosome FASTA, which is a tiny chromosome, dummy chromosome that we built for this tutorial. And you also have a small set of repeat modeler families that were found from that chromosome. So how do you navigate from one folder to the next? That is the command CD, which is change directory. So if you are in the TE man and not directory, you can do CD into bin, and that will take you to the next directory. And I think you have been shown how to list files within these directories. Okay, now it's the live demonstration. So I'm going to switch to my terminal now. Right. So I am a bit cheeky because the CI align modifications that I'm going to show you today are not 100% live. So I'm going to show them, but then you might have to wait a few days until Kathy, who is in the audience, I think, online, might be able to make those available for you. But I do have them installed in my laptop. However, the first round of just generate the priority table and make an alignment from the BLAST, we're going to be able to run it together. Yes. Oh, yeah, sure. Thanks. Is that good? Even more, perhaps? Yeah, OK. So as you can see, I've already been playing around with CI Align today, and it was very, very refreshing. I'm going to show you the results, and they're really, really cool. The first thing we're going to do, though, is to generate a priority table from the repeat modeler output. So repeat modeler will give you quite a large collection of libraries of prospective TEs. But if you are interested in start curating, and if you're like fresh new to curation of TEs, you're better starting with things that have protein domains on them and that have a lot of copies in the genome, because it's much easier to visualize alignments. So in order to do that, we created a script called generate priority list, and it is found in the bin. So I'm going to do an ls in bin, and you can see it there. It's called generate priority list, and it is a shell script. So I'm going to run it using bash. So if I do bash bin, and within the bin, I'm going to go to generate priority list. And if I click enter without nothing, it will print the help. So the help, it's a little bit too busy, but let's focus just on these 4 things for now. It tells you the parameters that expect after you run the command. So here it wants the repeat modeler output. It also wants the genome that you use to generate that library. They want a PFAM domain, PFAMDB database. Why is this? Because it will translate all the nucleotide sequences in your repeat modeler library into amino acids, and it will going to look into the PFAM domain, and it's going to give you, it's going to count how many PFAM domains is in each of those sequences. Because the more PFAM domains it has, the more likely it is to be a real TE. Of course, it could have PFAM domains that are not TEs, but then that's a different step that you can do to check. And the last thing it has, because my coding is very basic, is just you need to tell it where the repo is so you can find the scripts. So we're going to run that now. The repeat modeler output is in the tutorial data. So if you type tutorial, or a little trick for those who are new to Unix, if you press the key tab, it will autocomplete for you, because there's that many options that you can put in there in that directory. So we can try it again. I can try RM2, and if I click tab, it will complete. So that's the first parameter. The second parameter was the genome. So I'm going to put the genome there. It's something called chromosome FASTA. The third thing is the PFAM directory, and that is, I think it's one above. Where is it? Oh, this is my computer, so this is somewhere else. Yeah, this is going to be a problem. Where is it? Goodness. Oh, can I run it in the... Yeah, good point. I cannot even see. There we go. Documents being PFAM. Thanks. There you go. And the last thing is the GitHub repo, which is where I am at the moment. So I just can put a dot there. And this should run. So it's running CD-hit first. So it's taking the repeat modeler output, and it's trying to cluster those ones based on some parameters. That is all in the publication. You can check for that. So this can take a little bit. While I'm doing these, are there any questions? Yeah, thanks, Jessica.
For the PFAM database, it was just a directory. Are there specific files that need to be contained within that directory?
Yes, so the information for that, it's in the supplementary material of the paper.
Okay.
And it gives the paths to the PFAM files. I understand that now PFAM is being migrated to EVM. Yeah, so that might represent a few problems, but as soon as the migration happens, I need to talk to Rob about that, ask him where he's going to put them. We're going to update the links into sort of have an update on our GitHub.
Great, thank you.
So it's finishing running CD-hit, and now it's running PFAM. That's also going to take a little bit. In my other desk, I had everything run before, but then you can't. This is running real. Are there any questions at this point?
Hi, and this priority list that you're going to create, is there any way to, I don't know, change the priority order? Maybe if I'm more familiarized with certain kind of TE to start with those first?
That's a good question. We can actually go to that list. I think I have a slide that shows that. Yeah, so this slide has a snapshot of the priority list. Let me show it. Did it show? So I hit it. Yeah. So the priority list looks like this, and it has the name, the original name of the family. It has the length. It has the number of good blasts. So you can change that to whatever sensitivity you want. And it has the number of PFAM domains at the end. So you can sort them based on, we can sort that list based on whichever parameter you want. If you want to start with the longest ones, you can sort your table by the second column. In the bottom panel, I'm sorting by the number of PFAM domains, which is the last column, column 4. And I'm doing that -n -r says treat this as a number and do it in reverse order. So then you have at the top the ones that have the more number of PFAM domains. So LTR1 family 10 has 6 PFAM domains in it. So that's probably complete.
As an aside, if you haven't read the paper, that, you know, it's all of those like commands, sort, K, grep are really well explained. Supplementary data.
Yeah, no knowledge of units or units is expected to do this. You can start from scratch.
Is that? Oh, I forgot my question. Oh yeah, is it? Are you doing auth prediction or is it just like 6-frame translation? So
6-frame translation.
Yeah. Okay.
Yeah.
Great.
But yeah, that's it uses get auth. If you have frame shifts.
So I guess all the things will potentially be missed, or if they're a bit degraded. Or...
Yeah, but you know the script is just a bash script, and it
yeah.
has embos on it. So if you prefer to run transect rather than
transect,
just change it,
just plug it out.
Yeah,
okay. Yeah, cool.
it's very easy to get to.
Yeah.
Right. Hopefully this has finished. Yes. So it's finished. It says that it's finished. The PFAM searches, the priority table is generated. We can look up there, and that's the priority table at the end. And if I just do a final, then there it is, and it's the same as you've seen before. Okay, so that's the priority table. I would recommend you start with the ones that have more protein domains and have more blast hits. And of course, 1, 5, and 10 is going to be because it's a tiny little chromosome that I generated from this demo. If you have a full genome, some of them might only have 10, and I'm sorry about that. But some of them will have 500, and that would be a really good place to start alignment. Maybe choose 50, about 500. Okay, so let's see where we are. So what's the second demo? The second demo is to do make a FASTA from BLAST or make an alignment from BLAST. So what we're going to do now is we're going to use one of those high priority possible families to fish out the sequences in the genome and to create an alignment. So to do that, I'm going to choose one. I've already worked with one already that gives a really good result. So I'm just going to show that, and it's LTR1 family 4. So I'm going to look for that. So where is that family? That family is in the original repeat modeler output that you used. So we're going to use the command grep to find that particular name. I'm going to copy part of it. I'm going to change because I know it's 4, so I'm going to change that, and I'm going to use it. I'm going to search for it in repeat modeler to multi-FASTA file. grep is an expression matching sort of script. So it will look for that particular string. We call this a string, this collection of letters in this particular order. So I see we can find it. It's there. Fantastic. It found it. Now the problem is that that's only the header of the FASTA. There's no sequence in there. The sequence is probably on the second line. So what I can do is I can ask grep and say, okay, well, give me the second line as well as the one of matching. Oh, okay, well, give me a lot more. Oh, it's a minus A1. No, I think that should work. Yeah, okay. Okay, so he gave me the name, and then he gave me the first line. I'm pretty sure that's not the full sequence of that family. So, you can say, okay, well, give me the second one, but we cannot go one after one. So what can we do? Well, let's change this FASTA file into something that doesn't have ends of lines at the end of each sequence. So there's a script for that, don't worry. So I think it's a Perl script. So we can look into the bin, see what we find. So in bin, there's something that says FASTA multi to one line. And it gives you all the directions of what you need to do. So it says file contains a FASTA file. It says it will make this into this other thing. So it changes that into this. So when you have extract things that match a particular name, you can do it much easier. So we're going to do that with the repeat modeler one. And I'm going to put it into, I've already done it, so it's going to go fast. Okay, that was really fast. And now if I list on that particular file, you can see that they are all in one line. Yeah. So now I can go back to my grep script and replace the file that I'm looking, where I am looking for this particular family. And there is, oh, I got something else on there.
You had the picture.
Oh, I see. So now I can redirect that to a file I'm just going to call query. Okay. So now I can use that query to run that fast blast search onto the genome and make an alignment. Okay, that's a bash script. It's in bin, and it's called make alignment from blast. So this is going to do that part that I showed you earlier, which is this one. It's going to do this part. It's going to take that query that I gave it, that I'm going to give it. It's going to look for those locations in the genome. It's going to extend as many bases as I tell it to extend. And then it's going to lift them. And in this particular script, it's also going to run maft, which is one of the aligners. Okay. So let's do that. So this is, again, if I just do return there, it will tell me all the instructions that I need to know. And it's going to, it expects that I give it the genome, so the FASTA of the genome, the FASTA in, that is the query, the minimum length of the sequences that you want to lift from the genome. So this could be 0, and if you put 0, it's going to automatically get half of the sequence of your query. So if the query is 2,000 bases long, it's going to make that minimum length for the lift to be 1,000 bases match. And then the flank is how much do you want to extend to the sides. So the first one, we can run it with 0, so it's half the length of the query. And I'm also going to run it with 0 flank as well, so you can see what comes up. Okay. Right, so I need to give it the genome, which is here. I need to give it a query. I'm going to say 0 and 0. So that's going to run just a fast, and then it does the alignment. And I already have the results. I'm going to show it to you here. So this is something you don't have in your virtual machines. This is a genome viewer. This is GelView. There are a number of different flavors. GelView is one. AliView is another one. You can choose. This is extremely good to yeah, just have a look at how your alignments look like. So this is the one without any extension, and you can see that it has pretty good sharp borders. So that's the the right end. This is the left end, pretty good. But is it all? Is that the border of the TE? We can't know that. So we can extend and try to find out. So rather than do 0, I can do 50. That's going to run again. That's the blast. And then that's the alignment. And now I can look at, I can open the file. And it's what time is it now?
304.
I think it's this one, yeah. Okay. So this one has some more sequence on the side. It's also picked up a bit more of things that don't align very well. But this block, I'm not entirely sure that's the end of it. Not entirely sure. And let's look at the other end. The other end looks pretty conserved, and this one is the case extending it, but there's probably more sequence there. So we can run this again with maybe 500 now. So now we're extending even more and more and more. And you'll see that the alignments get a little bit more difficult to run. We can open the same sequence now. What's this one? Changing the color. Can I make this bigger? I don't know. And now, this, you see how this is just, there's no alignment. I mean, there's no conservation there. So I've definitely gone over the limit of my TE. And I know that because I've already done it, but I'm going to show that to you. So I can start walking. Oops, that's where it starts. I'll tell you a secret. There's a TG there. So that looks like the start of a LTR. So this is where, in our paper, we tell you that you need to manually curate this. So what are you going to do? You're going to crop this end. You can remove all those trailings of non-aligned bits that are not conserved. You can remove these indels here all manually. Or, with the new developments, we can use CI align and do that automatically. So I'm going to show you how to do that. So my colleague, Kathy Brown, who I think she's in the Zoom audience, has developed this tool with one of the PhD students in the group called Charlotte. And Kathy is still actively working on this. Oh, before I move into CI line, I want to, this is the advertisement part of the talk. So these are the supplementary materials of the paper that we've written. Includes videos on how to do manual curation of the alignments. And I mean, this is me talking to my screen and telling you what columns to delete and where the border ends. And I have to learn that from scratch. And I just hope that nobody has to go through the painful experience that I had to go through learning this remotely, which was, it was very good to get people from the community teaching me how to do this. But I hope this can get people started on that path. And the supplementary material has a large collection of protocols, everything from how to get started with installation of these tools, where to get your first steps in Unix, what is awk and grep and all these things, but also has things a little bit more, what's it, like advanced, yeah, there are more protocols in there that are not shown right here. But yeah, it's very basic. So. So CI line. CI line was published this year or last year, authored by Kathy Brown, who's the senior author in this paper, and it's a very user-friendly software, but with a bunch of different flags. You can just select how much you want to trim from each of the ends of your alignment, what is the minimum or maximum length of the deletions, insertions that you want to get rid of. This was not initially developed for TEs, but as I said earlier, Kathy and her team have developed a set of parameters that work really well, and there might be some ideas to try to develop this specifically for TEs. So I'm going to give the live demo of CI Align. And I am following Kathy's advice on this. So she very helpful in providing parameters that we can use. They have tried them out. So we were here, right? This is where we were. And we want to get rid of all these, we know the conservation stops here, starts there. So we want to get rid of everything to the left. We want to get rid of all these insertions. And there's a lot more stuff in there that we want to get rid of. There's a huge amount of insertions. And as we go to the other end, there's also more lack of conservation. And this is fine because I was trying to extend to find the border of that element. So it's okay. We can use now automatic tools to trim them. So that's the last alignment that I've generated as the one that I showed you on the viewer. So I'm going to run CI align for this one. I know this is an LTR already, so I know which flags to use. So CI align, I'm using the developer bit. Okay, the help of CI align is a little bit intimidating. It's very long, but also it's very well explained. It's long because it's very well explained, and each of the different flags are explained very carefully. So spend a little bit of time reading the paper, try to do the demos that they have, and spend a little time reading the help. Read the docs. So we've already pulled out a set of parameters that work, and they are here. And I'm happy to share these ones later on. Just drop me an email or find me on Slack. And I've had tried other things before. So yeah, so I recommend that you have another file where you can keep doing these things and keep changing them. So we're gonna crop divergent, and there's some of them can choose variables, so you can change these numbers to be more or less restricted. We're gonna remove short sequences, and that is a default of about 50. Then you can also, for example, determine what is the maximum or minimum insertion size, sizes that you want to allow. So I'm going to run these for our alignment. So I'm going to do CI align in file is the query that was the alignment. I'm just going to paste everything else about. I also added the last one is visualize, and that is important because then you get all the beautiful plots that I'm going to show you in a bit, and it shows you the input that you put, what modifications have been done, and also the way it looks in the output. It can also predict consensus sequences for you as well, and give different weights on types of consensus that you calculate. So this is also going to take a little bit of time. So I'm just going to jump to how the results look like. So that was the original alignment, say, for example, that we put in, okay? And remember that our query was only a fraction of that alignment, okay? So we did a blast, we extended, but now it looks very messy on the edges, and there's a lot of indels. So with CI align, with just one command line, we can switch from the top alignment to the bottom alignment. And that looks a lot better in terms of conservation. This is an LTR element, or it could be also a DNA. So these are the ones that you have borders, very clear borders at both ends. Line elements and Penelope present a little bit of an issue because you have this sort of, some of them are going to be shorter and all bigger because of the transposition and machinery. But it is also possible to move, transform using CI align, a very messy alignment, or, well, it's not messy, but it's just trying to be comprehensive and try to trim it to just the key aspects of it. So that is what one of the examples that we gave. We know this is a line family. After just one command line with the right parameters, and let's bear in mind, all the parameters that I just say the right parameters, that's going to be the same for all lines and for all Penelope. So once you work out those parameters, you can keep using, you can run these on a loop. You don't need to figure out for each of your lines, okay? You're going to end up with this lovely alignment. And as you can see, of course, this looks, ideally you want to look at it and order them from longer to shorter, and you will see that sort of inverse triangle. But if you look carefully, that is already there. A lot of them are full length, some of them are a bit smaller. But that consensus is going to look pretty good. So now back to our real life experiment. It's telling you what it's doing. It's removing the divergent sequences, doing all these things. So now it's just plotting the mini alignment. So it's just making those lovely figures for you. It also creates a fantastic log file when it tells you exactly what it's done. It tells you which column is deleted and why. So you can also, this is amazing for reproducibility, because one of the major problems of manual curation is that each person is going to do it different. If I give a family to each of you and I tell you to manually curate it, at the end, we're going to struggle to find two that are identical. If you do it like this, you're always going to get the same thing. So it's highly reproducible. So I'm going to, I know I'm pressing for time, so I'm just going to end up in a high. I'm just going to show you the results of our experiment. Here are all the files that I told you about. The remove txt, it tells you exactly what it's done. This is the clean, faster alignment. And this is the one that I'm going to open. And I was very happy when I did this earlier today to see that the first base was a T and the second one was a G. So you can have, I'm sorry, that it's probably a bit small for you, but you can see that pretty sharp TG there. And if I jump, you see all the insertions were removed, all the gaps were removed. There's some small sequences, perhaps there's some gaps. And at the left end, we have CA. The figures that you have, okay, so that's the input. That's the legend. That tells you exactly what it's done. So it shows you the bits that it has streamed are in pink, and that's the output. So this is in case you want to kind of quickly go through your figures and see what it's done. And finally, after you have your lovely alignment and you create a consensus, then you have to check that you have all the structural domains that that require for a particular domain. And this is a tool that was developed by Clem, and it's called TE8, and it has its own tutorial, but he was very, very generous and put it as part of our paper. And I cannot recommend this highly enough. It has 4 plots here. It tells you, so it takes your consensus and it remaps it to the genome. And here on the top left, you have the coverage of all the hits in the genome. On the right is a representation of the same. And this is very important. So the one on the left bottom is a dot plot. So here's where you can see the structural variations in terms of these sequences, the same as this sequence. And this is the typical plot of an LTR retrotransposon. And it also looks for conserved protein domains, and this is not PFAM, so this is complementary to the stuff that we were doing before. And finally, so the acknowledgements, I'm right on time. Oh no, I'm doing this 2 minutes fast. So yeah, so the first thanks goes to all the people that contributed to this paper. Kati, who is a great developer, has done this amazing tool, and she's very keen on learning from you what is necessary in terms of what can be improved to accommodate CI aligned to TEs. Christian Chaparro is my collaborator in France, and with him we've done the curation of these weird parasites. I need to give a massive thanks to the Slack community, both TE worldwide and Alex's Jamboree. My funding is from the Department of Pathology at Cambridge and at Christ College. Let's take questions. Thank you.
So I'm pretty sure I asked this question before and I forgot the answer. So when you now have these beautiful cuts like the LTR alignment you showed, the very beautiful, nice terminal sequences, is there some way to parse out the directly adjacent, in this case now something that's 4, 5, 6 base pairs long on both sides, so you can check for TSDs and lengths and motifs in there? Something that is it that works? I think I asked you this before. I forgot what you said. But...
So I think I asked you that question a while ago. Okay. So I suppose I don't know. I haven't spent enough time thinking about it. I would imagine you.
Yeah.
There you go.
There's a script part of TE tools. It's
yeah, that's right. We have a lot of
part of
TE
tools. It's tsd.pl.
tsd.pl, yeah. Because you do have the flanking sequences still there, right? I had several comments, I'm afraid. So when you search with BLAST to collect copies, you get everything back that it matches, right? So programs like the PEEP model are trying to kind of pull apart related things and build separate models for each other. But when you go back to collect things that look like the model, you will get the related sequences back too. So you start mixing up things that are very diverse.
So the BLAST is very stringent.
Okay, so you're only looking for very recent. You're working on very recent.
It's stringent, but for example, I can find DNA transposons in my parasite that are dead.
Yeah.
So it's I think I don't know if it's, but also this is something you can go into the. It's just a BLAST command in the bash. So if you wanted to make it more stringent, just increase the e value and you can make it lower. I would say yes, we're looking for things that at least like show some. But yeah, I mean, that is tweak, and I take that as a feedback. Yeah, thank you.
Yeah, so it's great that you have a seed alignment. We call these things seed alignments at the end. But Robert gets totally freaked out when you clip out the insertions, because then the sequence of the seed alignment does not match the original genomic sequence anymore, right?
Right, but
visually it is nice to cut them out. But when one would submit this alignment, we'd like to have those still in there, because they're being mapped back to the genome. So suddenly they're different because you clip them out of the multiple.
Right, but I would be under the impression that repeat massacre could deal with that and just.
This is if you want to submit it to DFAM.
Yeah.
So if you want to submit a seed alignment to DFAM, you shouldn't remove sequences from the seeds, because then it cannot be mapped to the genome anymore. This is,
Okay. Okay, we can take that out.
Hey, no one can hear you.
So you can remove the indels or leave the indels there.
Yeah.
CI align allows you to do both things.
Yeah, that's just, I understand that
Yeah.
it's possible. And visually it is nicer to have those because it gets really long after a while.
Yeah, so I guess one of the applications of this is that if you just, for example, in my case, I wanted to know how these look like in my parasite. So I wanted to see what protein domains were there, and in terms of, what the average, is there a real ORF there or not?
Yeah.
And so then like a very large insertion, if I want to create a consensus out of that I can pull out an ORF, I probably don't want that. But I can take it
but
that
it is not, those inserts are not going to be in a consensus.
Yeah.
They are, I just actually saw her in the chat, they're not, they're a minority. Yeah. I got more, but I think I'm hungry.
I think we're actually at time.
Yeah.
So to take the next coffee break, happy to coordinate in terms of like submitting data to DFAM. If you have any questions, obviously, Anna, very knowledgeable and can answer probably any questions that you have.
Also, Kathy is also now part of the TE Worldwide. Her name is Kathy Brown. So if you have any questions that are specific about CI line, I'll recommend you to direct them to her, because she's the person who developed it and can help you better than I can.
Well, let's give Anna a round of applause.